about the mid-1970s, most endocrinologists believed that peptide hormones bound their receptor, which are in the plasma membrane of the cell, exerted their biological action, and then came off the cell. We attempted to repeat such experiments using iodinated EGF and cells in culture. We found, certainly, that the iodinated EGF bound to receptors on the cell surface. Eventually, the radioactivity came off, but the hormone itself did not come off. What came off was completely degraded hormone. And we came to the conclusion that the hormone does not simply bind its receptor and come off, but that the hormone and the receptor go into the cell, and the hormone is eventually degraded. This was a new concept, and we thought it was necessary to prove this by other methods than using iodinated hormone.
He labeled EGF with fluorescein and saw it first as a halo. Then, sure enough, inside the cells. At higher magnification, electron-dense ferritin was used as a label. Here, the molecules show as individual dots, which are taken up by the cell as the plasma membrane invaginates. After 20 minutes, the EGF could be seen well inside the cell. Shortly after this, it disappears. Cohen's group wanted to look at the biochemical events associated with this internalization process. They used radioactively labeled ATP and looked for protein phosphorylation. Particularly strong label was associated with a band of 170,000 molecular weight.
The final conclusion from this and many other experiments is a new mechanism of hormone action. The EGF receptor is a molecule of around 170,000 glycoprotein. It sits through the membrane, part of it facing the outside of the world where it can bind EGF. The inside of the molecule that's facing the inside of the cell going through the membrane is a kinase. When EGF bound to the outside, the kinase domain on the inside of the receptor became more active and could phosphorylate not only substances in the cell, but also itself. So the receptor for EGF had at least 3 domains: an EGF binding domain, a catalytic site for transferring phosphorus from ATP, and a site that became labeled with the ATP. Well, that was very interesting because this was a new mechanism of hormone action where the receptor is the enzyme, one molecule with 2 heads.
In 1986, Cohen won the highly prized National Science Medal for his 25-year study of epidermal growth factor. A field which he started in 1962 with the discovery of a material which promoted cell maturation has since grown into an international research industry. The reason for this is simple. The idea that a simple hormone receptor system is a key switch for cell growth has powerful appeal for those looking for a cancer cure. As for understanding how the switch works, though, we still have a long way to go. Stanley Cohen may not learn these answers in his research lifetime, but in the next program we look at how other workers have picked up the challenge. Today, a biochemist's best friends include monoclonal antibodies, enzymes which slice up DNA into gene-sized bits, and other enzymes which stick them together again. This partnership is allowing us to learn much more about the EGF receptor and its role in cell proliferation.